Hey guys, you should have just finished kind of going over an analogy of the Mona Lisa leaving the Louvre and how important it is that it stays in place because it's such a valuable piece of art. And it's really the same concept with DNA, in that DNA is invaluable. If it were to leave the nucleus, the chances of it getting damaged are really likely. So the fact that it stays inside the nucleus makes it safe, and it is able to have a copy of it made in order to leave the nucleus, and that's how we are made. So basically, we're going to look at these components here. We've got adenine, thymine, guanine, and cytosine, which we can just go ahead and call A, T, C, and G from this point on, but they do pair in a very specific way. Thymine should always pair with adenine, and cytosine should always pair with guanine. So what they'll do is DNA is a double strand, and we've got one strand that will go on one side and the other strand goes on the other side, and that's called the complementary strand. So you'll see here I've got a C paired with a G, T paired with an A, and so on and so forth. It's always going to pair that way, and that ensures that the information is correct no matter what strand you're reading. We're going to go ahead and kind of practice what that looks like. So right here I've got a single strand of DNA, and it's comprised of those 4 letters. And you'll see here that I've got a letter 3 and a letter 5, and that actually tells you what direction to read it from. So right here, this 3 stands for 3', and that's where you're going to want to begin to read. And then we have the 5', and that's where you'll end reading. But a complementary strand is going to run backwards to that. So I'm going to go ahead and practice with you guys. If I've got a 5' strand here, and on this strand I've got an A, what would pair? It would be a letter T. And where I've got a T, I'm going to have an A pair. And where I've got a C next, I'll have a G. And then I've got the G, so I'll have a C, 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 G, T, A, T, C, G, and I'm going to go ahead and end that with a 3'. And that would be the proper way, and this would be a complementary strand to the strand that we just looked at. Does that make sense? Hopefully it does. If not, do a couple more practice problems. But if you'll see here, the way that they pair is very, very specific. The pink, which is representing probably the G, is always going to pair with a C, and the purple is always going to pair with this blue, or green, sorry. You'll see that represented throughout the entire strand. The fact that they're paired together, that they're linked together, those 2 complementary strands, by hydrogen bonds is a really unique thing. Hydrogen bonds are actually the weakest type of bonds, and this is important because usually being a very weak bond is a bad thing. In this case, it's a great thing because they're such weak bonds that they can come apart really easily, and when they come apart, we can actually read the information that's inside. And reading that information is inside is how a copy is made. That copy is called RNA, and what it looks like is this strand right here. So if you look at the differences between the two, this is DNA, it's a double helix, which means that we've got these sugars and phosphates making up this solid line on the outside, and then these little ladder rungs on the inside are made up of adenine, guanine, thymine, and cytosine. And so what you'll see here is very, very similar, except for the fact that it's single-stranded. It only has one strand. It's leaving the nucleus, and so it doesn't need to be extra safe. As soon as it's turned its information into something else, then it's going to go ahead and be destroyed. But you might notice that we've got the color yellow, which is represented on there as well, and we've got the color blue, which is represented on there as well, and we have green, but we do have something different instead of red. And so what that's going to indicate is on DNA, wherever we have a thymine as a base, that doesn't exist as far as RNA goes. It's actually going to be called uracil. So wherever you would have an A pairing with a T, that A is instead going to pair with a U when it's on the uracil, when it's on the RNA strand. What happens is this blue is representing DNA, and you can see that it's a double helix, but you can also see that it's unzipped here in this little bubble region. And the fact that it unzipped is because of those hydrogen molecules just breaking apart because they're very weak. And when they unzip, we've got this molecule called RNA polymerase, and it comes in, it latches on to the DNA, and it just starts making a copy. So wherever there's an A, instead of bringing a T, it'll bring a U. Wherever there's a T, it'll still bring an A. Wherever there's a C, it'll bring a G, and a G, it'll bring a C. And in doing that, it makes an exact complementary copy of that DNA, which can now leave the nucleus, travel through the cytoplasm, and find a ribosome. If you remember from our unit on organelles, a ribosome is where proteins are made, and proteins make you. So it's really important that it's able to do that and make that copy. This process is called transcription. Transcription literally means to write. So if you think about that RNA molecule and that RNA polymerase, it's literally writing down a copy of the DNA, and that written copy can leave the nucleus. It's like hearing a recipe from somebody and then writing down that copy of a recipe. Now you can, leave their house or leave wherever you are, bring that written copy with you, and it can be translated into a recipe no matter where you go. The differences between, we looked at the picture, but here's some solidified differences. We've got the double-stranded for DNA, the fact that it contains thymine, and then it has a deoxyribose sugar. DNA stands for deoxyribonucleic acid. So it tells you that it's a deoxyribose sugar and that it also contains nucleic acids, which are those bases, A, T, C, and G. And the fact that it's only stored inside of the nucleus. Whereas RNA is a single strand, it has uracil instead of thymine, and it's a ribose sugar. It stands for ribose nucleic acid, and it can travel out of the nucleus and through the cytoplasm to find a ribosome. Kind of looks like that. And we're going to do a little bit of a practice. I wanted you guys to kind of look here. I'm going to erase this. We're going to do it together. This is the exact same DNA strand that we looked at earlier, and we made a DNA copy of it. But now if we're to make an RNA copy of it, we're still going to have a 5'. And here where I have an A, there's not going to be a T, right? There's going to be a U. But I have a T here, so I'll have an A with the C. I'll have a G, C, 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 G, U, A, U, G, C. And then we're going to end that with a 3'. I keep doing that. Okay, so I want you guys to kind of finish this up, and you're going to do a couple practice problems on the next slide. Make sure that you understand how to do this and that you're comfortable with it.